Hello guys, how are you? So in this lesson we shall study about gene interaction. This course is presented by me, Navneet Sharma. You can know more about me from here. And I would also love to tell you that I have recently cleared a URATPG entrance examination with All India rank 13. You can follow me on this link, and if you like the lesson, kindly rate, review and recommend. Now beginning with the lesson, Non-Mendelian Inheritance. One character may be influenced by many genes, or many characters are influenced by a single gene. The Mendel's theory states that each character is determined by two genes. It is believed that single phenotypic character depends upon the combined action of genes and environmental actions. Okay. Now, many examples can be quoted which deviate from Mendel's principles. These are called as the non-Mendelian inheritance. In some examples, the genes may interact in different patterns. This concept was introduced by Bateson, and it is known as Bateson's hypothesis. Now, the complementary genes 9:7 ratio. These genes are two pairs of non-allelic dominant genes which interact, produce only one type of phenotypic character. Like in man, deaf mutism is an example of complementary genes. In normal hearing and speech, in man develops as a result of interaction between gene A and gene B. Whenever a person is homozygous for either of the two recessive alleles, that is either capital A, small b, or small a, capital B, he is a deaf mute. Okay. For example, you can refer to this diagram. This is a dihybrid cross of deaf mute person. That is, we have these parental. Now, and these are the gametes produced, and when these gametes are crossed, in some of the cases, deaf mute person may be produced. Okay. So moving forward, supplementary genes 9:3:4 ratio. These are the two pairs of dominant alleles which interact in such a way that one dominant allele will produce its effect whether the other is present or not. The second dominant gene, when added, changes in the expression of first one, but only in the presence of the first one. Okay. Now in rats and guinea pigs, the coat color is governed by two dominant genes, that is capital A and capital C. The agouti colored guinea pigs have genotype capital C, capital C, capital A, capital A. The black mice possesses factor for black color, that is 6, but not gene, okay, for agouti color. But if gene for the black color is absent, agouti is unable to express itself, and mice with genotype small c, capital A are albino. Here the presence of capital C produced black color, and addition of the gene capital A changed its expression to agouti color. Okay. So this is an interesting type of genes. Now dominant epistasis, 12:3:1 ratio. Epistasis, that is the called as act of stopping, is the interaction between non-allelic genes which are present on separate loci, in which one gene suppresses the expression of other gene. The gene which suppresses the other is known as inhibiting or epistatic factor, and the one which is prevented from exhibiting itself is known as the hypostatic gene. Now, although it is similar to dominance and recessiveness, but the two factors occupy two different loci. Okay. Therefore, while dominance involved intragenic or intraallelic gene suppression, the epistasis involves intergenic suppression. Okay. And now there is another example. Parental generation shows a brown dog and a white dog. These are the gametes formed, and in the F1 generation, all white dogs were formed. When these were crossed, okay, in the F2 generation, we obtained this result, okay, in which 12 were white, 3 were black, and 1 was brown. Okay, so you can study in detail from here. Now in dominant epistasis, the two pairs of genes, the dominant allele is gene A, or one masks the activity of other allelic pair. Okay, Since the dominant epistatic gene A exerts its influence by suppressing the expression of gene B, it is known as dominant epistasis. The example of dominant epistasis in dogs. In dogs, while coat color appears to be dominant, it develops due to the action of epistatic gene I, which prevents the formation of pigment controlled by hypostatic gene B. Now, this hypostatic gene B produces black coat while its hypostatic gene produces brown coat color only when gene I is recessive. Okay, only when gene I is recessive. Remember that. Now, the progeny of dominant I does not allow them to function and result in white coat color. Next, we have when these are the examples of that. Okay, when two white coat dogs are crossed, they produce white, black, and brown in the ratio of 12:3 respectively. The white dogs in this case possess gene for black or brown coat but does not produce pigment because of the presence of gene I in dominant state. Now, when two pairs of genes determine the same or nearly same phenotype, so that either of the two genes is able to exhibit character in question, these are said to be duplicate factors for the seed pods. For example, fruit shape in shepherd's purse. This is a shell found in shepherd purse. That there are two types of seed capsules. That is, first one being triangular, and second one can be elongated or top shaped. These were when crossed, they produced triangular and elongated fruits in the ratio of 1:5:1 respectively. Okay, sorry, if it is 15:1 respectively. Now the triangular characters of seed is controlled by two pairs of genes, capital T and capital D, which are present on two different loci. Okay, remember that they are on two different loci. It is not the case of dominance or dominant or recessive. Okay, in the dominant or recessive case, the genes were present on the same loci, but in this case, these genes are present on different loci in two different chromosomes. Okay, now both of them either collectively or singly produce triangular characters or pods. Elongated pods appear only when these dominant genes are absent. Such genes are called as duplicate genes or pseudo alleles. Okay, these are called as pseudo alleles. Okay, now thus the Mendelian F1 ratio, that is 9:3:3:1, is deviated in different organisms in different conditions, and the genes interact and exhibit deviated ratios in the F2 generation in many cases. Okay, so this was the introduction of genes of non-Mendelian inheritance. So I hope you like the lesson. Thank you guys for watching. Keep learning.